Thank you very much, Sangeeta. That was a very kind introduction. Pam and Jim, what a day. This is an amazing conference. The excitement about science, seeing all of this here, and then people staying around. This is something that I know it took a lot of work. I'm going to be talking about this one checkpoint, PD-1 blockade, and trying to understand how it works. And I'm going to be quite reductionist. It's going to be simple concepts that drive the efficacy. Here are my disclosures. I've worked with companies, big and small. The only one that would be relevant, it's called Pac Pharma, which is a company there. But unfortunately, it's not a disclosure anymore because the company no longer exist. If we think about a graph like this one, where we have here Jim's funding discovery of CTLA-4 limiting immune responses to cancer and how we can release it, we can think of if somebody like me, a clinician, gives a PD-1 blocking antibody, we're making one important assumption. We're making the assumption that there's a whole bunch of T cells that had gone through everything else that's required to induce an immune response, and they're stuck in this last checkpoint. While they're trying to attack the cancer protects itself. This is overall in patients with cancer not common. When it works, when you release PD-1 and the patients respond, they go on to do very well. But it's something that has to be regulated, that this is not a patient is lucky and responds, or a patient is unlucky does not respond. There has to be a series of rules that allow the immune system to do this. And I'm going to go through these 4 very simple rules based on analysis of patient biopsies and conducting clinical trials. The first one is very logical. The immune cells have to recognize the cancer cells different from normal cells. That's logical. And the more different the cancer cells are from normal cells, it will be better to work. And here we have this spread of mutational load between different cancers. And Bob Schreiber and Don Schumacher separated them and said the ones in green that have the high mutational load are the ones that are more likely to respond to PD-1 blockade therapy. And some of them are colorectal and uterus. There's a group of them that are very high. Those are the MSI high colorectal and uteral cancers. There's a group of melanomas that have very high mutational load. That group of melanomas was a subset of melanomas called desmoplastic melanoma. You may have never heard of desmoplastic melanoma, or if you've heard about desmoplastic melanomas, because I don't stop talking about it, because I think it's a very interesting cancer. Desmoplasia is the pathological term for fibrosis. And I'm sure most of you have gone to immunology conferences and heard that fibrosis is bad for the immune system. Well, a cancer that's defined by fibrosis is the one that I want to portray. Also, it's dedifferentiated. It's not even pigmented. It's closer to the neural crest than melanocytic. But it is a cancer induced by the carcinogenic effect of this ultraviolet light and has a mutational burden that's 10 times higher than cutaneous melanomas. So with my colleagues at the Southwest Oncology Group, an NCI-funded cooperative group, we said, let's test if the good features, more mutations, overcome the bad features, fibrosis and dedifferentiated. So we did 2 groups of patients, and we gave them anti-PD-1 either in the neoadjuvant setting before surgery or in advanced disease. And the data is amazing. Patients who had 3 doses of pembrolizumab before surgery, who had biopsy-proven desmoplastic melanoma at baseline, 71% of them had no cancer cell left in the surgical specimen. In metastatic disease, we had an 89% response rate, and here we have the waterfall plot. And this is a cancer that used to be among the worst and the worst. Here we see a PET scan at baseline of a patient with multiple metastases, and here we can see a PET scan after treatment. The tracer it shows the brain, the bladder, and in that case the heart. So that's not melanoma. That's just an earlier scan. You can see these systemic responses. And in patients with localized disease, we can have cases like this one. This is a patient with a lip melanoma that was biopsied, and the surgeon was planning to cut 2 centimeters on each side of the lip, reconstructed with the skin from another part of the body, and that was within a week of when I saw the patient. And so the patient, well, let's see if this is a desmoplastic melanoma, and if it is, I have a clinical trial for you. And I was so sure that the patient would respond, because by then I had seen enough of these responses, that I asked the patient, take a picture of your lip every day and send them to me. And the patient took a picture every day. Here you can see day 3, it's already smaller. Day 7, it's smaller. A week later, you can barely see it. 2 weeks later, you can barely see it. 3 months later, I send the patient to the surgeon. The surgeon, I don't know what I'm going to resect. I didn't tattoo it. I forgot what it was. So this patient never had surgery, responded to anti-PD-1 therapy. I also like showing this because there's another concept that I hear in many conferences, which is pseudo-progression and inflammatory responses. I don't know where you see the pseudo-progression or inflammation. It doesn't even get read. The T cells go in, kill the cancer cells, and killing the cancer cells leads to not having the tumor microenvironment. The fibrosis goes away, everything else goes away. So you need to have a very good T cell response for this. We'd entered 55 patients within both cohorts, neoadjuvant, so surgically resectable or advanced, and we looked at the 3-year melanoma-specific survival. That means looking at how many patients died from melanoma. And actually, there's only been 1 patient out of 55 who died from melanoma. Another patient died from a metastatic cancer that was not biopsied. These are exceptional responders. I know it's an extreme example. There's another extreme example, the MSI-high colorectal cancers with Miriam Chalabi and John Hannon here, and then Andrea Churchek and Luis Diaz. These cancers are poised to respond because they have high mutations and the immune system is ready. And I see my friend Jen Wargo here. I'm going to be a little bit provocative because it didn't matter what these patients ate. It didn't matter if they were on probiotics or antibiotics. It didn't matter if they got their infusions in the morning or in the afternoon. And what the heck, it didn't matter if they got the COVID vaccine or not. They all responded. So if you have a highly immunogenic cancer, it's likely to respond. The opposite is the same, the truth with it. The cancer is low immunogenic. You can pile up immune checkpoints. You may never get a response. So that's on one side, but you have to have the immune system recognizing the cancer. And that is not that simple either, because our T cells have the T cell receptors that are recombined randomly to recognize the universe of potential things. But they had to be pre-existing. And when we started with this over 10 years ago with Paul Toomey, and when at the time when you could publish a paper in a major journal without doing single-cell sequencing, we just did simple immunohistochemistry and look for, are there T cells there? And if they're there, those patients are likely to respond, and that's on the top. And on the bottom, there's 24 patients here. You can barely see the lines. But you can see that the big difference of the presence of T cells that are the ones that are going to be responding to these therapies. And if we get a little bit more complicated, we do immunofluorescence for PD-1, the receptor, and PD-L1, the ligand, and they have to be in physical contact. It's a logical thing. This is an antibody that was designed to displace this interaction between PD-1 and PD-L1. And if you don't have T cells that recognize the cancer, good luck trying to block PD-1 and getting a response. But the key interaction is between a T cell receptor that recognizes a peptide presented by HLA, and that is a difficult thing to study. We can simplify things and look for the extreme examples, but the majority of cases will be wondering what is in the cancer that's being recognized by T cells. And the problem with studying this is that we have 10 to the 15 possible T cell receptor sequences, and over 24,000 HLA class 1 alleles have been described in humans. We can get the problem smaller. There's 65 HLA class 1 alleles that would cover 99% of the population. So we could have templates for those HLA alleles and be able to try to study immune responses. And we did that with working by Jim Heath, who was at Caltech at the time, and then Institute of Systems Biology, and David Baltimore, obviously at Caltech. And we developed approaches to be able to sort T cells based on what their T cell receptor recognizes. And following a little bit of what Kathy presented this morning, the process is to look for the differentially expressed mutated peptides in cancer cells and make single-chain trimerase made out of a library of 1 of 65 HLA class 1 alleles, have the DNA sequence with a linker with beta-2 microglobulin that's required for MHC class 1, with a linker and the peptide sequence that comes from the mutation. And I'm seeing Elaine Mardis in the audience, the first one who described this. And we did this academically, and then we created this company, Pac Pharma, and Pac Pharma would do 350 of these soluble reagents that will be multimerized, barcoded, and then co-cultured with T cells from patients, so we could then study the landscape of T cell responses to mutated peptides presented by the patient's HLA class 1. And we wanted to use this so we could study patients who responded or did not respond to PD-1 blockade therapy. What are their T cells recognizing? And here's work that we did with Pac Pharma, who was then my postdoc, Chris Puch-Saus, and now is a faculty member at UCLA. Sequencing tumors from 11 patients who received anti-PD-1 therapy. 7 of them had clinical responses, 4 of them did not have clinical responses. We tried to see, using these technologies, how many mutations are recognized. And we can go from 3,500 mutations that only 11 of them are recognized. Here, 900 mutations in patients without a response, 4 of them were recognized. This is a phenomenon called immunodominance, where you have potential recognition of multiple antigens, and the immune system converges in some. That did not differentiate the patients with a response or not response. It was a handful of mutations that were recognized. What separated the clinical responses to PD-1 blockade or not were how many T cell receptors were recognizing these mutations. Patients without clinical responses had fewer T cell receptors than the patients who had multiple, that had clinical responses. And you may continue looking at these, and these are just bubbles of data. I'm going to show you that the T cell receptors that we cloned from these patients would recognize the cancer. And we used these cases because we had made cell lines from their melanoma biopsies, so we could clone T cell receptors that we thought they were recognizing, and then go culture them. And so to do this, we had to develop an approach to to insert T cell receptors. We did this with Alex Marson some years ago. We would, in one step, knock out the endogenous T cell receptors and put the transgenic T cell receptors, and then be able to test the T cell receptors and see if they kill cancer cells. The melanoma cells are in red, and the T cells on top clear them up. Here's one of the T cell receptors. This is patient number 2. We clone 14 of these T cell receptors. I'm showing you the killing of one. You may think, are you showing the best one? Well, no, because the other 13 kill exactly the same. So all of them had this activity once they were selected this way. And even in patients who did not respond to anti-PD-1 therapy, we still could find some T cell receptors on this patient number 1, one T cell receptor. Here there's 2, there's 2. So it tells us that even in patients who do not respond to anti-PD-1 blockade with just one agent, we could probably reverse the resistance by giving more immunological therapy. And we did that by saying, well, what if we try to reverse resistance by giving an earlier checkpoint, CTLA-4, and then to see if we have to give it alone or in combination. And to do that, we did it again with this cooperative group where we took patients who had metastatic melanoma, were progressing on anti-PD-1, and we gave them either EP-Nivo or EP alone. And the results showed that in a third of the patients, we could reverse resistance to anti-PD-1 by adding anti-CTLA-4. And it was the combination that was needed, not CTLA-4 alone. So anybody who has tried to look for antigen-specific T cells, good luck trying to find them because they're rare, either in blood or in tumors. And they don't like to be in the environment that they are. So there has to be something that helps the T cells do the job. Here, I'm just making a depiction of the tumor microenvironment. There's a T cell seeing antigen, but the tumor microenvironment is full of cells that are trying to not get T cells to work, including Tregs. And it has the wrong pH. If you try to culture T cells in this environment, they don't like to proliferate there also. Something needs to change. What changes is that the T cells recognize the cancer, and by recognizing the cancer, they start making interferon gamma, and interferon gamma forces the cancer cells to be better recognized, to lead to the production of more chemokines that attracts more T cells, and those T cells make more interferon gamma, and the cancer cells are now starting to make 1,500 interferon-stimulated genes within hours of being recognized, and that keeps spreading in the tumor, and we know that it goes up to 40 cell layers because Ton Schumacher did an experiment where he put an interferon gamma reporter gene, and you could see that you could turn on genes 40 cell layers for where T cells are making tumors, are recognizing the tumor. So by getting here, any of you would say, well, yeah, Tony, you've gotten really good at biorender and you spend time making cartoons, but can you demonstrate that this is right, that this is true? And I'm pretty sure that Phil Sharp, if I gave him enough time, you say, well, why don't you do the genetic experiment? Why don't you, if you're trying to convince us that interferon gamma is doing all of this, you knock out the interferon gamma receptor or the signaling and then see if this happens. And Phil, we've done the genetic experiment in mouse models and in tissue culture, but unfortunately, cancer has done this, melanoma has done this in patients. And it's going to be hard to go through this without having a pointer. But here's two of the patients that we treated years ago who had response to the phase 1 of pembrolizumab and then progressed. And we sequenced the resistant tumor and compared it to the baseline tumor. And we did, meaning Jesse Zareski, did these circus plots. And there's no way I can go through these by myself. So I'm going to come down. Can I have you as a volunteer? I'll make you look good. Yes, sure. What's your name? Alejandro. Can you come here? So that side there is a circus plot where we're comparing the resistant tumor from the baseline tumor. And we organize the mutations where the shared mutations are in gray, the lost mutations are in green, and the new mutations are in red. We're going to look for a new homozygous mutation. And I'll tell you what we're looking for. New homozygous mutation would be where you see where BAF1 says. You see, but if you go around, there's a white line. If you go up on chromosome 1, do you see a red dot in BAF1? Yes. You see 1. Yeah. So Alejandro, can you go around all of the circle on that? Do you see another red dot on the BAF1? Bearing a little... No. Nope. So Alejandro has gone through 22 chromosomes, 20,000 genes, that has found only one new homozygous mutation. The one new homozygous mutation was a JAK1 mutation. Let's do the other one. I already gave it up. Let's go to the BAF1. Let's go all around. Do you see a red dot?
Yep, down there.
You see one red dot. You go around.
Only one.
Alejandro, thank you very much. He's a very fast learner. The second one, there was only one new homozygous mutation. That was a JAK2 loss of function mutation. There's no way this can be by chance. I'm presenting this not because it's frequent, I'm presenting this because I think it's important. And if we map these two kinases, JAK1 and JAK2, they're the required kinase for the interferon gamma receptor to signal, where you have them, they phosphorylate starts and they turn on these 1,500 interferon-stimulated genes. And the lack of this these kinases leads to the not expression of these genes, which leads to this acquired resistance where now the tumor can grow despite T cells being able to recognize it. So I think this cartoon is actually right. All of these may be okay descriptive science. I'm just trying to describe what happens when the therapy works and what happens when it doesn't respond. But can we turn the knowledge to be able to change treatment? And actually, if you have to have pre-existing T cells in the tumor, we can think of changing the paradigm of care. And this follows what Pam Sharma pioneered years ago, which is the standard of care is to do surgery where you take the tumor, and with the tumor you take a whole bunch of immune cells because they're attracted there because the antigen is there. And then we give immune checkpoint blockade and we hope that the patients do better, and actually they do better in here. But what if we change the sequence? We do Pam Sharma's neoadjuvant, and then we turn on the T cells, and then we let Jen Wargo take the tumor out, but we've already turned on the T cells, and then do we have a better outcome? We proposed to do this study to the NIH, and the statisticians told us, this makes no sense. You're giving the same treatment to two groups of patients, 18 doses of pembrolizumab and surgery. There was one important difference, which is the surgery was placed at a different time so we could turn on the immune system before surgery. And actually, the clinical trial was amazing. The neoadjuvant adjuvant group had close to 50% lower event-free survival than the adjuvant group. This, we generated this with Sapna Patel, who was at MD Anderson. There's been a lot of investigators at MD Anderson that pioneered this. And similar data has been generated by Christian Blanc and Georgina Long with 2 doses of ipi/nivo before surgery, turning on immune responses before the surgery. So with this, what I wanted to convey to you is that PD-1 blockade is a very targeted immunotherapy. It only works when there's a certain biology, when the T cells had recognized cancer before and they were stopped by just one thing. So I've gone over some of the people who worked on this and helped us along the way from my lab and other collaborators, our colleagues from SWOG, the remaining people from my lab, and since I became a prelude to Fred Ramsdell, I'm glad that Fred and Laura came down from the mountains. Yes.
Thank you, Tony. I think we will have time for one question.
We're maybe late in the day, so we'll have it at drinks.
That's fine. Thank you, Tony. And with this, we conclude session 3. And I would like to thank all the extraordinary speakers. And I would like to say that your pioneering work would keep redefining the field of cancer immunotherapy. Thank you. Video.
The closing keynote address will be given by Fred Ramsdell, Ph.D., founder of Sonoma Biotherapeutics and the 2025 Nobel Laureate in Physiology or Medicine, recognized for his pioneering work on immunoregulation. Together with Mary Brunko, Dr. Ramsdell identified FOXP3 as the molecular determinant of regulatory T cell function, uncovering mechanisms fundamental to autoimmunity, cancer, and immune-based therapies. Please welcome Dr. Fred Ramsdell.
Thank you. Thank you. And we would like, it's a pleasure to welcome our 2025 Nobel Laureate in Physiology and Medicine, Fred Ramsdell. And thank you so much for putting immunology on the spot one more time after Jim. Thank you.
Thank you. You're the first person to officially congratulate me, so that's awesome. I want to thank Pam and Jim, except Jim left. But he's with the band. He's tuning up. I'll try to get you out of here so you can hear the band, and I'm guessing there are drinks involved. I'll try to get you out of here on time. But I do want to thank Pam and Jim and the rest of the Institute for inviting a random immunologist as opposed to a cancer immunologist to this august day. It's been fantastic. The speakers have been amazing. The science is just wonderful. And I will say that Pam was one of the first people to call me, and without a doubt, the most enthusiastic person to call me. Far more enthusiastic than I was, actually, and I was pretty happy. So if you don't want Pam to call you all the time, do not let her have your cell phone number. So I'll just say that. It's just why mine is usually on silent or airplane mode. Okay, so somebody mentioned one of the earlier talks, and I've forgotten who, that they used the phrase back in the day. I'm going to take you way back in the day. So you're going to have to indulge me. There was no single-cell-seq. There was no multi-omics. I know that's the topic, but that didn't exist. So I'm going to do a little historical walk for everybody, and if you don't find it interesting, you can leave, I suppose. So I'm going to do two parts of this. I will actually, at the end, kind of bring it full circle, which is amazing, although I'm going to skip 20 years in between, and you'll see what I mean in a second. So, and these are my disclosures. So, a lot of people don't even know that this place existed unless you saw the Manhattan Project, et cetera. But this is Oak Ridge National Labs. This was built during World War II. And it was built principally to make the bomb. It made the atomic bomb. Now, the US government decided it would probably be good if we had some idea what all that radiation was going to do to mammals. So they also set up a mammalian genetics unit. But the principal use was this, and you'll see up here, it says, it's hard to read, but it says, what you see here, what you do here, what you hear here, when you leave here, let it stay here. So people in the nearest cities didn't even know this place existed. It was so secret when they built it. It's kind of crazy. But they set up this mammalian genetics unit, and it was headed by Bill and Leanne Russell. And this is in the 1940s. And I was noticing the mouse cages haven't really changed all that much since the 40s. But they set this up, and kind of a funny story is Bill and Lee would take cages of mice, put them in the back of their Volvo, drive to White Sands, New Mexico, where they were testing atomic bombs, put them at varying distances from the atomic bomb, go underground in a little shelter, bomb goes off, gather up all the cages of mice, drive back to Oak Ridge National Labs, and see what happened. And so that's actually how we figured out radiation dosage for people. It's kind of crazy. The mouse I'm going to talk about never left the facility in Oak Ridge. It's a spontaneous mutation. Kind of crazy. But serendipity is important. So this is sort of the timeline for this. And you can see, Manhattan Project starting in the early 40s. World War II hadn't ended, right? DNA structure we didn't know for another decade. It took a long time. The scurfy mouse I'll talk about came along in the mid-40s. And so you can see that there was some dashed lines in there because not really very much happened for a long time, except that they maintained this strain of mice, this line of mice, for many, many years. So the other co-laureate for this year is Shimon Sakaguchi from Japan. And in the mid-90s, early to mid-90s, he described this population of cells, which he referred to as regulatory cells, which if you remove them before birth or right after birth, animals got sick, and if you isolated them and put them back in, you could decrease autoimmunity. But it was a pretty phenomenological thing, and it came on the heels of a lot of suppressor cell work, which people decided was not real, which was largely true because it was sketchy. But these cells were still very interesting, and we identified the gene in 1998, and I'll tell that story right now. But again, just for reference, this is before the human genome was sequenced. So sequencing was still a really big deal back then. This is the types of mutations that you could generate using the ionizing radiation. I don't even know where I got this cartoon anymore. And it's politically incorrect, and I just bear with me. But the scurfy mutation was a spontaneous recessive mutation. It was X-linked, so half of the male animals would come down with this disease. And it was a pan-autoimmunity. So these animals develop diabetes, they develop psoriasis, skin manifestations, they develop cardiomyopathy, inflammatory bowel disease. These animals all die by about 3 weeks of age, and there's really not much that keeps them alive. And the people at Oak Ridge kept this strain going for 300 generations and tried to understand it as best that they could. And again, this is all, by the way, with your NIH tax dollars, just so you know. That's just not true. This was Department of Energy tax dollars, but let's not split hairs. So when we got the mice in Seattle, we developed a collaboration with them, and the tools had changed in the mid-90s, and we said, we can find this gene. So when we got the mice, we characterized them, and again, there's a lot more data. I'm just going to throw up a couple of pieces here, but we showed, and Lisa Clark was a postdoc in the lab at the time, that these animals were hyperproliferative. So if you tickled them at all with an antibody to the T-cell receptor or CD3, et cetera, they would divide and they would make a ton of IL-2. They didn't care about co-stimulation. This is when Jim was describing co-stimulation as being really important, and I'm like, not so much. So they went crazy. They would go crazy. They didn't care about co-stimulation in the slightest bit. I can say this because he's not here. So we decided to map this. Oh, can I go back one? Sorry, I just needed to show Mary's name. So I had the great fortune to work with a woman named Mary Brunko. She's still a friend. She was a tremendously talented, molecular geneticist and mouse geneticist, most importantly. And Mary came to this little company called Darwin Molecular. That's not our official logo, but it's cuter than our official logo was, so I put it on the slide. Company's gone. I don't think they can sue me. So she came to this little company that was started in the Seattle area, and there were probably 30 or 40 of us at the company when we started this. And we looked and said, 3,300 kb, that was the region that this phenotype had been mapped to. Well, that was too big to sequence back in the day. I'll use the phrase back in the day a lot. So that was just too big. We couldn't do it. So we had to do a backcross. We didn't have a mouse facility. So we're really interested. We know this is really important. We'll do a backcross. So we took over the janitor's closet. So we put a set of mouse cages in the janitor's closet. I don't know what the janitor did. I never asked. And we just made a mouse room out of it. And we picked the janitor's closet because it had a sink so we could wash the poo out of the mouse cages. It's awesome. So we mapped it and did a backcross and got this down to about 500 kilobases, which was still a lot in 1996 or 7, whenever this was. But we decided we could sequence 500 kilobases. And we knew there were basically 20 open reading frames in that 500 kb. Okay? And some of these things, I mean, they've got numbers, right? No one knew what those were. And so, okay, that's fine. So we have to start somewhere, so we prioritized, and I prioritized this list. And remember, I'm working for a company, and what our ultimate goal is to find novel things that we can utilize to develop drugs to treat human disease. And so whatever this gene was responsible for pan-autoimmunity in this mouse. This has got to be important. Okay, but we got to be able to drug it eventually. So I prioritized this, and when I could figure it out, I tried to find things that looked like maybe it had a transmembrane domain or something. It was hard to tell. And we looked for expression of all these genes in various tissues, and it was not good. You did it by northern blots back in the day, and it wasn't particularly helpful. But we started sequencing, went through all of them. Now, that took a little while. So in 1998, that took us 3 months, which we thought was actually really fast. Today, it will take you less than an hour. But then it took us 3 months, and that was great. But we kept going through genes, and we never found the gene. And you start to lose hope at some point, or at least I did. We got to the last gene, and one of the guys walked into my lab and said, "We got it." And it was this gene called FKH. You'll notice it was literally the last gene. I could not have been more wrong about prioritization than I was. So now, no one should tell the Nobel Committee that, but I could not possibly have been more wrong. And so he brought it in and he dropped this notebook on my desk, and he pointing to the two little base pair insertion in there, which ultimately led to a premature termination and a non-functional protein. Great. This was April in 1998. And I looked and I was like, this is awesome. This is great. Crap. This is a transcription factor. Now, in the year 1998, transcription factors were not druggable. They may be druggable today, but they were certainly not druggable in 1998. And I was like, well, what am I going to do with this? So there was this elation and this oh my God, this is a terrible target. What do I do with this terrible target? So it was kind of a mixed blessing at the time. I think I used a lot of other stronger words than crap, actually. But so that was fantastic. However, what do I do with it? I didn't know Andrea Scheitinger. Actually, I think she was in elementary school when we did this or something. So that wouldn't have helped me very much anyway. But okay, so I've talked about that. I'm going to ignore that. But this is what the gene looked like. So it had a fork-head binding domain, winged helix, fork-head binding domain. So it's a DNA binding domain. It's got a leucine zipper and a zinc finger. Helps for dimerization with a number of things. This is old. If you're taking pictures of this is probably wrong now. But I'm using old slides, so there you go. And then it has a repressor domain. And it was pretty clear this is probably going to be a transcriptional repressor. Great. Still not helpful to me, because I've got no way to use this particular discovery, which was, again, exciting, but at the same time, really pretty frustrating. So the gene got named FOXP3. You did not get to choose the name. They gave it to you. There's a nomenclature committee. You did get to choose the name of the protein, however. So we called it scurfin. We thought that was a nice name. No one ever uses it, but I still like it. So that was really nice. We did the formal proof to say, all right, is this really the right gene? So we wanted to complement it genetically. So at the time, we made back transgenics because we didn't know enough about the gene regulation, et cetera. So we put a 20 kb back into mice. And you can see the gene itself is about 8 kb. And we also made, I won't show the data, but we also made animals in which we only expressed this gene in the thymus. They got disease like the wild-type mice. So you had to have this gene on both in the thymus and in the periphery, which was, we weren't sure how that was going to go. So that was pretty interesting. So we made the animals. This is one of these moments where this is the proof. Like, this has to work. And so Mary and I basically sat down, and we decided we were going to dissect the mice because we really wanted to know if this was right. So you've got this is the lymph node from a normal mouse on the left, the scurfy mutant mouse on the right. It's big. They often get bigger. It doesn't show so well here. And a mouse that is genotypically scurfy, but also carries, in this case, 5 copies of the transgene, and it looks like a wild-type animal. These mice live just fine, perfectly normal, and so we knew we had the right gene at this point. So we were pretty excited. So what else do we know about these animals? We know we need to press the button. There we go. So then we, of course, said, well, that's great for the mice. What about people? I don't need to cure mice. That's not important, or at least not as interesting as it could be. So we collaborated with Hans Ochs at the University of Washington and Bob Wilden at Oregon Health Sciences and Mark Appleby, who was in our group. And we identified several and I realized the image is over the text, but it doesn't matter. They identified several populations of individuals in which there was a familial disease in which half of the male children died usually before 10 years of age. And so we said, okay. So we got DNA and we got as many as we could from within the family. We began to sequence. And there were we looked at about half a dozen families early on. And it was very clear that the lethal phenotype correlated with mutations in FOXP3. And in fact, every change that at least as of, a few years back, every change in an amino acid in FOXP3 is pathologic. So nature does not screw with this disease, or with this gene. There's not a single amino acid that's different, which is kind of remarkable to me, actually. I was very surprised. But there's about 200-ish, maybe 300-ish families in the world who have mutations in this. And so now people are actually trying to put the gene back in and do gene rescue to treat the patients here. So that was great. So we also studied the mice. And so the transgenic animals, I said, so you can see on here, this is just the proliferation assay to titration of T-cell stimulation. And the scurfy mice is the top thing. You can see that they respond at very, very low doses. Wild-type mice are in the middle. And the transgenics, they weren't wild-type. They were actually hypo-responsive, which was not something we expected. And that was a very repeatable finding, and it was confusing for quite a while because the cell numbers look normal. The cells generally look normal overall. And so Rolly Kotri, who's on the slide, and she's actually the first person in the world to know that FOXP3 controlled Tregs, and you'll see why here in just a second. I mentioned that we looked at expression of this gene by northern blot at one point, and this is obviously much more recent data, but the gene is pretty ubiquitous if you look at a tissue level. Over there in the green on the far side are lymphoid tissues. It's no higher than anything else. It's pretty ubiquitous everywhere, and that's one of the reasons it was at the bottom of the list. Mostly it was because it was transcription factor, but it was also ubiquitous. And I thought, well, this is an immunological problem. It can't be in every tissue. That doesn't make any sense. But it is, and it does make sense, of course, once you actually get it. And the reason is, it turns out when you look at hematopoietic cells and lymphoid cells, this little population of cells that's circled, which is 10% of CD4 T lymphocytes, is the population that Shimon Sakaguchi, the other person with which we share the prize, had described as being these suppressor cells, these regulatory T cells. And so in mice, they form basically a very distinct population of cells because mice aren't typically responding to pathogens all the time. They're sitting in an SPF environment, which is a little dirty, but not really dirty. So they have this little population. It's really easy to find them. And so when you look at the gene expression, it turns out the gene is almost uniquely expressed in that population of cells. So we said, I still don't understand why it's in all the tissues. That doesn't make any sense to me. Well, it turns out regulatory T cells are in all your tissues. They hang out in the tissues. You have regulatory T cells in your muscle, in your fat, in your whatever tissue all the time. And they're there as a surveillance mechanism, and they stay there all the time. They traffic in and out, but if you look at the repertoire of regulatory T cells from your liver versus your skin versus anywhere else, they're all slightly different. So the T cell repertoire is a little bit different because, presumably, the antigens that are expressed in those tissues are slightly different. So these things are tuned to tissues without any inflammation whatsoever. It doesn't really matter, which is why this gene is associated with all the tissues, because there's regulatory T cells in all your tissues. And those keep you from becoming autoimmune. I hit that twice. Okay, it doesn't matter. That's good. So we performed an assay that was messed up on the slide, but that's okay. Described by Angela Thornton and Ethan Shivak at the NIH. And they actually described this assay at the exact same time we were identifying FOXP3. In fact, I was a reviewer on the manuscript, and it was sitting at my desk, and Rolly came in and told me what she'd seen, and I said, do this assay, because we need this data. And so the assay here is very simple. Blue is simply responding CD4 T cells. So just your basic CD4 T cells. They're being stimulated in a variety of ways. The orange is those same T cells, but regulatory T cells added in. And so on the left part of this panel, it's just regular CD4 T cells from a wild-type mouse. If you add non-regulatory T cells, you get more proliferation because you've got more responding cells. Not surprising. That's how it should look. When you do it in the transgenic animals, however, now these other non-regulatory cells look regulatory. So we've enforced a regulatory program onto these other T cells. Now it's interesting, and it's potentially important, and it may be therapeutically relevant. I think we'll figure that out in a few years. But you can enforce that program on other T cell populations. If you put B cells in there, it doesn't work. So you can express FOXP3 in B cells poorly, actually, but they don't look like regulatory cells. So T cells will do this, which is, I was pretty surprised to see that, but it clearly worked. And if you overexpress this gene to significant levels, so we had some transgenic animals that had something like, I can't remember, 40 copies of the transgene, you could barely find a lymph node. There were very few lymphocytes left in these animals. They simply did not develop normal T cells at all. So, all right, so the first part of this, how am I doing on time? Oh, I got plenty of time. So the first part of this is, all right, so FOXP3 is the master regulator for Tregs. That was great. And it was a very nice convergence of the cell type that Shimon Sakaguchi had studied and characterized, and then our ability to find the molecular genetic basis for how those cells develop and how they actually work, it was fantastic. Clearly, absence of a functional protein results in lethal autoimmunity in mice and in humans. And I mentioned the thymic expression as well. So this really ended up being, and it is to this day, a necessary gene for maintaining peripheral tolerance. Clearly we have central tolerance. We delete most of the T cells that are responsive to our own body, but not all, as any person with an autoimmune disease can tell you. And this is one way of controlling those cells. So that was great. So even back in the year 2000 or early 2000s, we recognized this is great. So if we could give these cells to people, we could treat autoimmunity. If we could find ways to expand these cells in people, make them more active in people, et cetera, we could treat autoimmunity. The part of that was the challenge was if. And back in the year 2000, no one would think of doing cell therapy for autoimmunity. We didn't have really good drugs or ways to amplify these cells. We didn't, at that point, know that low-dose IL-2 might do that. And so, although it is actually on the slide. But we thought about that, but we didn't think there was any really good, viable way of making this work. And on the flip side, it was pretty clear that, you know, if these cells existed in tumors, and you know, other people have shown that to be true, if you can inhibit those, you've got a better chance of getting an anti-tumor response. So, 15 years go by. And a lot of people do a lot of work. We know a lot more about how FOXP3 and Tregs work, and that's fantastic. And so, so now we come to about 2015, and people began to think, all right, we can put cells into autoimmune patients. CAR Ts are working for oncology. You can grow these cells. It's a viable therapeutic approach. Let's do it for autoimmunity. And so the first studies were done. Well, let me diverge for a second. Forgot, maybe out of order. But one of the reasons that I personally like this cell a lot is I got a Nobel Prize. But another reason is it really is multifunctional. You're tapping into, you know, millions of years of evolution to get a cell that is, its entire function is to prevent you from getting autoimmunity. So I don't have to be smart. The cell is smart. So yes, anti-IL-17 works for some things, anti-TNFs work in some people, they work great. So there are targeted things that work really nicely in some patients, but it's still a minority of patients with autoimmune diseases. These cells know how to prevent autoimmune disease and control autoimmune disease. So my view was, let's use the cell. The cell has been generated through, again, millions of years of evolution. Let's not try to outthink evolution. So the first studies were done by Jeff Bluestone and colleagues, Qizi Tong, and other people have done very similar studies in which people grew up lots of these cells. And Jeff and others found ways to purify these cells out of humans, which is actually a little tricky. It's a little bit of an art. It depends on where you set the gate on the flow cytometer, which is a frustrating thing to try to explain to a contract organization. But you can do it. And they could grow them up, and they put them back into patients. You can get a lot of them. You can show that they're very active in all the whatever assays you want to run, whether they're in mice or in tissue culture lab. They're stable. They're epigenetically, they look like regulatory T cells. I won't talk about the epigenetics, but I think it's really important in this particular case. So they put them back in. Everyone was safe, no problems. You could, and I'll show you in a second, you could find them up to a year later in blood. That was great. You could see hints of biological activity in things like skin biopsies and other things where you could see interferon gamma went down in the biopsy a little bit. Okay, that's great. But there are no clinically meaningful responses in doing all of these studies. And so the feeling was we needed to engineer these cells and make them better. And we spent a lot of time debating what better meant, and I think we will still debate what better means. But that's where we're headed right now. I just wanted to show this because people ask about the stability of these cells over time, et cetera. And on the left side, you can see that, so these cells, this is a study from Jeff's lab, and they radio-labeled these cells with deuterium. And so you can see at 1 year, at a high dose of cells, now that's 3 times 10 to the 8th cells, or 3 times 10 to the 9th cells in some cases, you can still see cells at a year out in the blood, which is not really where these cells should be if they're going to be effective. So that was encouraging. And when you isolated those cells, and you isolated a bunch of other subsets, which is the other graph, the only place you could find the deuterium was in the regulatory T cells. So if these cells had lost their regulatory phenotype, if they de-differentiated, or if they were unstable, you would see them in other cell populations, and there was no evidence of that whatsoever. So once these are put back into patients, they seem to be incredibly stable. So Jeff and I and Qizi Tong and Sasha Radensky in 2019 started Sonoma Bio. And I will tell you, late in 2019 was a terrible time to start a biotech company because there was this pandemic thing coming on 4 months later, and we had to shut down the labs, and that was not fun. But what we have done is the approach here is to purify the same Tregs, very much the way Jeff did it in the polyclonal studies, engineer them in the lab, give them specificity, and in some cases, I think in future iterations, give them other properties, et cetera. Our first attempt on this is to go into rheumatoid arthritis patients, and there's a lot of reasons for that. There's a ton of preclinical data that suggests Tregs can reverse and inhibit RA in mice. RA patients have, by some measures, defective regulatory T cells, but when we take those cells out, grow them, expand them for 14 days in the lab, they look great, and they function really well. So that's not really a problem. They don't expand quite as well as healthy volunteers, but they expand sufficiently for what we need to do. We have inserted into them, is this it? Yeah, a CAR that recognizes an inflammation-associated antigen, so citrullonated proteins. So you may know that in rheumatoid arthritis, citrullonated antigens are a very common target for the immune system. Many patients have what's called ACPAs, which are antibodies to these citrullonated proteins, Vimentin being the most common, but there are many others. And so we worked with a group at the Karolinska to scan through a whole bunch of antibodies. We found one that seemed to see a variety of citrullonated proteins with good specificity and good affinity. So we've generated a CAR and put that into our Treg cells. And we can, and just to make sure we're dotting on the I's and crossing the T's, we can find citrullonated protein within the joints of RA patients. This is both intracellular and extracellular, so that's important for a CAR Treg. And in this particular case, even the synovial fluid, and it's not a dramatic effect, but it's real, even the synovial fluid will activate that CAR. So we think the synovial fluid actually has aggregates of citrullonated proteins, and that's why that works. But we can even show the synovial fluid will activate this CAR. And so this is what the CAR looks like. It's a typical antibody, transmembrane domain, and a couple of signaling domains. The domains are a little bit different in the context of a Treg, so it's not 4-1BB, it's CD28, because that works better in Tregs than 4-1BB does, for whatever reason. Well, for a number of different reasons. We also have a tag on this. And there's many other variations on this theme that we can do. Obviously, if we're going to engineer this, we could put in other genes as well. And those are certainly within the queue. But this is the first thing going into patients. I shouldn't say going into patients. It's actually in patients. I'll show you that in just well, I won't show you. I'll show you the protocol in a second. But the CAR works quite well on these Treg cells. So whether you're looking at proliferation, IL-10 production, other measures of Treg activity, it works quite nicely. It works so far in every product from a patient so far has worked quite well in this particular assay. And again, they're quite stable over time. You can see FOXP3 on the left here. Helios is another measure of an activated Treg. And Sonoma's developed a little fingerprint to compare regular Tregs with our transduced Tregs with T effector cells. And the cells that we generate look incredibly stable. They look stable epigenetically as well. Interestingly enough, and I don't know whether this will matter or not, but when we put FOXP3 into non-Treg cells, we can get some of this pattern, but not all of it. They do not look stable. They look somewhere in between a regulatory T cell and an effector T cell. So I don't know if that is important or not, but they don't look exactly like a regulatory T cell when you do that. Okay, and we've found a way to manufacture these things in a reasonable way, and it's very similar to what one would do for a CAR in oncology. And again, we have a fingerprint that we use to verify everything coming out of it. I won't spend a lot of time on this since I'm down to 1 minute. So, less than 1 minute. So we're in patients, 3 dose levels, and we've completed dose level 3. By we, I mean a lot of other people, because I'm not doing any of this. But completed dose level 3. The data is going to come out, I believe, next week. I can't tell you. I don't even know all the data, so I really can't tell you. But the data is going to come out next week. So we've been able to expand. So the safety committees look and make sure you can go up to the next dose level, et cetera. And it's a safety and tolerability study, as they all are. But of course, you're looking for activity. With cell therapy, this is a long, arduous, expensive process. We really want to see some activity in this. So I don't know what it's going to look like, but we'll find out very soon. So I'm going to finish. I just ran out of time. I'm going to finish with my acknowledgments. And so there's 4 people on this slide that I've highlighted. There's a whole bunch of people who contributed to this. David Gallus is one of the reasons we were able to do this. David passed away about 2 years ago from glioblastoma. And I reached out to a number of people in the network here to help with his care, and everyone was incredibly supportive and generous, which was amazing, because this is an amazing community. But David was a molecular biologist extraordinaire at USC for a long time, and then he ran the Department of Energy's biological programs. So he knew about Oak Ridge National Labs, and he knew the collection of mice that they had, and he could get us into that facility and create the relationships that we needed as a biotech company to get access. And David was a tremendous person. Steve Ziegler on the bottom was the first person who saw the scurvy mouse and went, that's interesting, we should work on that. And I was fortunate enough to be the one who worked on it. Mary Brunko on the upper right is the person who led all the cloning efforts, and Rolly Kotri on the bottom is really the person who ran all the biology. On the Sonoma side, Jeff Bluestone is a friend and collaborator for many years, and he's the head of Sonoma Bio, and when he said, I think we can raise enough money to start a biotech company to use Tregs, I said, when do we start? I didn't really give him a choice. He had to hire me at this, because I wasn't going to not be in on this process. Josh Bialke runs a lot of the research programs there, and those are other folks along the way who make this work. And of course, I don't have patience, so I got to thank the mice. So I tried to find the cutest mouse picture I could, and that's where I was. I got to thank the mice. Wait. 2009, first time I went to Stockholm. So we're walking down the streets in Stockholm. I realized I didn't have a lot of hair in 2009 either, but at least it wasn't all white. But we stumbled across this thing, and I literally said, this is the closest I'm ever going to get to the Nobel Museum.
Thank you, Fred.
Thank you.
Wonderful. So we will have time for 1 or 2 questions.
Or we can go drink. Sorry.
Or buy Fred a drink.
Oh, yes.
Or someone can buy me a drink, yeah.
Exactly. When we can discuss it during the reception.
Thank you very much.
Thank
it was
you.
lovely.
Thank you, everyone.
Thank you.
Thank you, Fred.
It was fantastic.
Closing remarks will be given by Elaine Mardis, PhD, Professor of Pediatrics at The Ohio State University College of Medicine, Co-Executive Director of the Steve and Cindy Rasmussen Institute for Genomic Medicine, Nationwide Children's Hospital, and co-chair of the Allison Institute Scientific Advisory Board. Please welcome Dr. Elaine Mardis.
Thanks, everyone, and thanks for the opportunity to be the closer here. I promise not to keep you from drinks and great music for too many minutes. But I just have a few comments to make. I guess first, from my own perspective, wow, just wow. What a day this has been. Yes. I just want to thank some key individuals and groups. So events like this don't just happen. I think we all know that if we've been involved in organizing these types of events. They take planning and they take a lot of effort behind the scenes and during the day for people to get up here, do their thing, and answer questions, etc. So I'd really like for us to first acknowledge those who had a role in making this symposium an incredible success, including the JPAI staff, the audiovisual team, the caterers, and our security detail and our volunteers. Let's give them a round of applause. Also, sincere thanks to Pam and Jim, somewhere out tuning up his harmonica, for your commitment to building this institute as a collaborative community within the MD Anderson ecosystem. Your tireless pursuit of excellence is the tide that rises all boats here. Congratulations. I also want to acknowledge the vision and commitment that was needed by Dr. Peter Pisters and Dr. Giulio Drietti to really get this institute established and really make the space that you're all in. Congratulations to them on this great success and their continuing support of it. Thanks especially to all of our speakers for their time and commitment, for sharing their science and their experiences for the benefit of everyone here. I think it's been truly inspirational to hear from everyone at this symposium. And thanks to the attendees for your interest and your desire to learn from the speakers and others who are here. And to everyone, I think here, I would like to reflect some of my favorite phrases that I heard during this time together. First of all, remember, science is hard. In spite of all the challenges that we're facing, science and medicine have enormous value. So stay the course, and above all, stay curious. And so thank you for listening. I'd like to now invite everyone to our post-symposium reception. It's in the park. If you need directions, there will be staff to point you in the right direction. I look forward to seeing everyone there. Thanks again and take care.